It is my great pleasure to introduce Karsten Borgwardt. Karsten first came to work with us at NICTA in Canberra. Was it in 2003? 2004. So I've known him for 7 or 8 years. Karsten received his PhD from LMU in Munich under the supervision of Hans-Peter Kriegel, Alex Mola, and myself. And he is now with the MPI. I always get this wrong. So he's with the MPI for developmental biology and MPI for intelligent systems in Tübingen. Karsten has done a lot of work on graph kernels before, and now he's moving more and more into working with real biologists. So he's one of the few machine learners who really works with biologists, shares office with buildings with biologists where they run around doing experiments on monkeys. Well, the biologists do that, but Karsten really is one of the few people who has an insider perspective on real bioinformatics problems, and today he is going to talk about how machine learning can be used in statistical genetics.
Okay. Thank you, Vishy, for the very kind introduction. And it's also my great pleasure to be here and to talk about machine learning and statistical genetics. As Vishy has told you, my own background used to be very method-driven. With Vishy, I've worked for years on the question of how to design graph kernels, so how to design measures of similarities on graphs, on networks. And with Alex Muller, I worked on designing statistical measures of dependence that use kernel methods to do so. When I moved to Tübingen, however, my research started to become very problem-driven. Now I'm working with my group on the topic of machine learning in statistical genetics. This is a very young subtopic of machine learning in computational biology. So you may have heard of the many different applications there are of machine learning in computational biology. You may predict the function of a gene, of a protein. You may predict whether a patient has cancer or not. So these are the topics that have been out there for a while and which are also very interesting. Statistical genetics has recently become one area in which machine learning can be applied. And why is this the case? Well, a major reason why there's now huge interest in machine learning and statistical genetics is that the techniques for measuring both the genotype and the phenotype of an individual have been improved tremendously over the last 20 years. The genotype of a person, that's the genome, that's the genetic information of an individual. And we now have all these sequencing techniques, we have these chip technologies that allow us to measure the genotype of an individual person, plant, or animal quite cheaply. The goal is that we may be able to sequence the full genome of a human rapidly and for less than $1,000. Currently, it's something like several thousands of dollars using the cheapest techniques. Well, humans is one species in which genetics tries to explore this connection between genotype and phenotype. Other areas include agriculture, where we try to explore how the genes of a plant determine its phenotypes. Phenotypes are the physical appearances of a plant. It's also called a trait. So it's a property of a plant that you can observe. For instance, one property of a plant that is very important is whether it grows quickly or whether it grows slowly. This is of huge agricultural interest to find out if there is a genetic basis for rapid growth in plants. Another question that is very important in plants is this plant resistant to some kind of bug? And this might also be genetically determined. Here on this slide, I've shown a few plants in which a lot of research is going on currently. In Tübingen, the plant genetics department is in particular looking at the model organism Arabidopsis thaliana. So this is less of agricultural importance, it's more of basic research importance. This plant, so a lot of basic biological mechanisms have been first studied in Arabidopsis thaliana. It's like the Drosophila of plants. And as Max Planck is an institute for basic research, our plant genetics department is focusing on studying the properties of Arabidopsis thaliana. And often they are the first ones to uncover new principles that have not been explored in other plant species before. Two plants that are of huge agricultural importance and for which there's also a lot of research going on are maize up here and rice. So recently there were papers in Nature Genetics where they published the genotypes of several hundreds of accessions, of several hundreds of individuals for rice and maize, and tried to find out which positions in these genomes determine whether such a plant, a maize plant or a rice plant, grows quickly, flowers early. Flowering time is a very important trait in plant genetics because it tells you a lot about how quickly the plant developed. We also work with researchers that focus on human genetics, on clinical genetics, with the MPI for psychiatry in Munich. So they are very much interested in the question, can you predict whether a depressive human patient responds well to antidepressant treatment or not? They have observed that when they give antidepressant drugs to their patients, then in 2/3 of cases, there is a reaction, so the patient gets better. In 1/3 of all cases, there is no reaction at all. Their conjecture, their hypothesis is that this might be genetically driven. However, this is not proven yet that this is genetically driven. Still, the techniques I'm going to present to you allow you to try to find out if this hypothesis holds, if you can find evidence that there is a genetic basis of for the reaction to antidepressant treatment. Well, I already mentioned that the genotypes can now be recorded at an unprecedented level of detail. The same holds for the phenotypes. By taking pictures of plants or humans, by taking videos, by recording time series of measurements, we can now get very accurate descriptions of phenotypes, which wasn't that easy, that fast, and that cheap 20 years ago. So what we see now in genetics is both on the genotype level and on the phenotype level, we have enormous amounts of data. And this is the reason why there is so much interest now in machine learning and in data mining in genetics, because they want to make use of these large amounts of data. They want to find patterns in these data. They want to find the statistical dependencies between genotype and phenotype. And this is exactly the classic question that genetics has tried to answer over the last 100 years. Now, to put the research question these geneticists are after into a more machine learning style of language, you could say that they try to predict the phenotype of an individual based on its genotype. And the iconic figure that is often used to describe how they approach this problem is shown here on this slide. So what a typical research question that you could ask is, you have a collection of plants that grow tall and short plants of the same species. Let's say these are Arabidopsis plants. And you have genotype information for each of these plants. So you know the genomes of, or at least parts of the genomes of these plants. And now you want to find out if there are specific positions in these genomes which control, which regulate whether the plant grows tall or whether the plant grows short. This is called a genome-wide association mapping. Genome-wide because we look at the whole genome, association mapping because we try to find out which positions in the genome correlate, are associated with our phenotype of interest. In this case, it would be the height of the plant. And now, the standard technique for approaching this in statistical genetics is that you compute some kind of association score, of correlation score between each position in the genome and the phenotype of all plants. So you try to find out if there are positions in the genome that have a specific nucleotide, so a specific letter in the genetic alphabet, that is always correlated with growing tall. Whereas there is another letter, another nucleotide, another base at this position when the plants grow short. And this you find out by computing this score of association. Now, how could you straightforwardly use machine learning in such a setting? Well, a straightforward thing to do here would be to try to predict the phenotype of a plant based on its genotype. And that's one of the first experiments we did in this direction. We looked at phenotypes from Arabidopsis from a recent Nature paper by Atwell et al., 2010. The Atwell et al. Study 107 phenotypes in this paper, out of which 9 are binary. And we examined the following 5 here in detail for 99 to 199 plants. And we had for each of these plants 250,000 SNPs. So 250,000 features. Each of these features describes one position in the genome. SNP stands for a single nucleotide polymorphism. So it's one position in the genome where different individuals have different nucleotides. So these are the positions in the genome where 2 individuals of the same species differ. So in this setting, we now try to predict, based on these 250,000 features per individual, whether this particular plant has a binary phenotype or not. So the first 3 phenotypes in this list here have something to do with the color of the leaves of a plant, and the latter 2 describe whether the leaves of a plant are rolled. And now, when we use a very basic technique, a support vector machine with a linear kernel on these single nucleotide polymorphisms in 10-fold cross-validation, then we get areas under the curve which are in between roughly 57% and 70%. These results are significantly better than random, but they are not excellent either. So they would be very good if we had at least 80 or even better 90 or more % of area under the curve. Currently, the levels that we observe are more around 60-ish. Now, why is there so much room for improvement in these predictions? Why are our predictions of Arabidopsis phenotypes only a bit better than random? There was a question?
Do you have any estimates on the heritability of these phenotypes?
So these are conjectured to be highly heritable, unlike many clinical phenotypes where we don't know the exact level of heritability. But these are highly conjectured to be genetically inherited. Well, why is there so much room for improvement? There are a lot of reasons why there is so much room for improvement. Of course, it could be that these phenotypes are not fully genetically determined. There may be gene-environment interactions, for example. So the environment may have an effect on these phenotypes. You can already see that this could be the case when looking at this table. So the biologists measured these phenotypes at different temperatures. So there's already a hint that they might conjecture that temperature plays a role in whether these phenotypes occur or not. And the technique we have used here does not consider these interactions between the environment and the genetics of a plant. What we also don't consider is the so-called population structure. So it may be that the plants which have a phenotype, which for instance have rolled leaves, are more closely related to each other than the plants that do not have rolled leaves. This may cause us to, and the support vector machine, to pick up positions in the genome that are shared between all these related plants. These positions in the genome have nothing to do with this particular phenotype. They just show up because the plants that have the phenotype are more closely related to each other than they are to the plants that don't have the phenotype. So this population structure describes systematic ancestry differences between cases and controls, between plants that have the phenotype and plants that don't have the phenotype. There's a lot of effort and a lot of interest in correcting for this population structure in statistical genetics. And for instance, Elazar Eskin at UCLA and others are working on this problem. Also, Christoph Lippert from my group has recently published a paper on this topic of how to correct for population structure. When we use a linear kernel here for predicting the phenotypes, then we make another assumption which may be unrealistic, which is that the effects of the different SNPs, of the different positions in the genome, are additive. So we do not allow for any interactions between positions between SNPs in the genome that may affect the phenotype. SNPs affect or may affect the expression of genes, the activities of proteins. Hence, we ignore any gene-gene interaction effects that may have an impact on our phenotype in this model. Now, why do we do this? Why don't we consider these gene-gene interactions? Well, considering gene-gene interactions, and in particular SNP-SNP interactions, is a very difficult problem. Why is it so difficult? Typical data sets that we use in genetics nowadays include 10 to the 5 to 10 to the 7 SNPs per individual. So if we want to look at all pairs of SNPs in these data sets and find out if they are associated with the phenotype, then we have to look at order 10 to the 10 to 10 to the 14 pairs. This leads to two problems. One is we have an enormous multiple hypothesis testing problem. If we check for each of these pairs, if they are in association with the phenotype, then we are testing order 10 to the 14 hypothesis. If we do not correct for multiple hypothesis testing, then by random chance, we'll get positive results for order 10 to the 12 of these pairs. So one challenge here is to correct for the enormous number of hypothesis tests that we are performing. Another challenge is the enormous runtime that this requires. We are performing 10 to the 14 tests, so even a huge computer cluster can be kept busy for several days computing all of these association scores for SNP pairs. In my talk today, I want to present an approach to you that we recently developed to overcome this computational runtime problem in epistasis detection. It will be at the core of the remaining time of my talk. And in this contribution, we assume that our phenotypes are binary. So as before, we have cases and controls. We assume that genotypes may be homozygous or heterozygous. And we assume that there are m individuals with n SNPs each. And what we recently discovered was an algorithm to find SNP pairs that interact with the phenotype in a runtime which is subquadratic in the number of SNPs. So we found an algorithm which is provably better, faster than the exhaustive search. this term epistatic interaction here, I would like to explain that. That is just the same thing as a SNP-SNP interaction. Epistasis is the name for the phenomenon that two genes interact with each other to cause a certain phenotype. Now, we were not the first ones to look at this problem. There are many other techniques in statistical genetics that have been used to solve or to tackle this problem. The first and obvious one is to use an exhaustive enumeration of all SNP pairs. However, here you either have to limit yourselves to hundreds of SNPs, or you have to resort to special hardware, such as graphical processing units. This is actually one approach that we also worked and work on, and I'll come back to that later at the end of my talk. Now, if you really go to a genome-wide scale, where you have 10 to the 5 or 10 to the 7 SNPs, then exhaustive enumeration without special hardware is not an option. Therefore, so-called filtering approaches have been developed. They are also called two-stage approaches, which describes the way they work quite well. In a first step, you reduce your set of SNPs by a filtering criterion. In your second step, you then just look at those pairs of SNPs where each of the two SNPs passed the filter you defined before. How do you define these filters? Well, you can define them statistically, or you can define them biologically. A common statistical filter, statistical criterion that is used to reduce the set of SNPs is to consider only SNPs with a large main effect. And by that I mean SNPs that on their own have a large association score with the phenotype. So SNPs that on their own are highly correlated with the phenotype. As a biological filter is typically defined via biological networks. Here, you only look at those pairs of SNPs that are in close proximity to genes that are interacting, that are known to interact from the biological literature. So that's the biological way of filtering SNPs and of reducing the number of pairs that you look at. In data mining, there were recently new approaches defined for this problem. Two famous ones are FASA-NOVA and TEAM. Here they use branch and bound techniques to prune away parts of the search space, so to avoid to look at all SNP pairs, or to efficiently update the contingency tables which they need for their statistical tests when they do permutation tests in order to compute p-values for measuring the statistical significance of an association. Now, in my group, as I said before, we try to work problem-driven. So we did not just use our method of choice for this task. We tried each of these different types of approaches. As I said, we tried the exhaustive enumeration, but using a graphical processing units. I will come back to that later. We also tried the filtering approaches, both using biological prior knowledge, compiled by a postdoc in the plant genetics department. He compiled a network of genes that interact in order to regulate flowering time in Arabidopsis thaliana. And we reduced the set of pairs of SNPs that we search for, or which we search in Arabidopsis thaliana when trying to map a flowering time phenotype using this gene network. However, what we observed here is that by just focusing on SNP pairs that are in proximity to genes from this gene network, we lost something like 80% of all of the most interesting SNP pairs. So imagine that you compute your test statistics for all your up to 10 to the 14 SNP pairs. And then what the biologists usually are interested in are the top 100 pairs in your ranking. And when we use this biological filter, we lost up to 80% out of these 100 pairs. So we had a dramatically low recall rate in many cases. We also tried a statistical filtering criterion, a machine learning based criterion where we used a support vector machine. We tried to find out which SNPs allow you to predict the phenotype with a decent level of accuracy, and then we only considered the pairs of SNPs that allowed you to do so. The problem here was that these phenotypes are usually not completely penetrant. Penetrance means if you have the genetic information in your genome that may cause a disease, then it's often not 100% sure that you will actually get the disease. So there is a higher risk that you might get it, but there's no guarantee that you will get it. And as soon as we moved to such models where there is so-called incomplete penetrance, so where the probability of getting a disease is even not 100% when you have a disease-prone genotype, then this approach failed and we couldn't get good results. We also tried index structure-based approaches, so we defined our own branch and bound-based algorithms based on Pearsons correlation coefficient. And here we first observed that this actually worked quite well. We could prune away 90% of the search space in our first data sets that we tried. However, when we moved on to more data sets, we observed that this pruning power is highly variable. So sometimes we could prove 90% of the search space, sometimes we could only prove 1% of the search space. So these approaches here, from our experience, sometimes do not prune the search space very well, and then they approach an exhaustive enumeration. Now, after all these failed attempts, we tried to tackle the problem using techniques from theoretical computer science. And actually, a bachelor student, Panagiotis Arlioptas, who visited Tübingen with an Erasmus scholarship, worked on this problem. And to find this theoretical computer science approach, we first had to decide how exactly we would like to measure the association between a SNP pair and the phenotype. Because there are many different ways of doing this in the literature, you could use something like Pearsons correlation coefficient, you could use something like a square test, you could use a regression model. So there are many different ways of how to measure the association between the genotype and the phenotype. The criterion that we agreed upon in the end is shown here. It's the so-called criterion of difference in correlation. As stated before, we assume binary phenotypes, cases, and controls. And we now try to find the pair of SNPs for which the difference in correlation between cases and controls is maximal. There's a very plausible biological interpretation of this criterion, which is that there's different degree of linkage disequilibrium between these two loci in cases and controls, or in less biological words, if this criterion here is large, then the two loci, the two SNPs, tend to be co-inherited either in the persons that have the disease or in the persons that don't have the disease. So this difference in correlation is a good measure to quantify whether certain SNP pairs tend to often co-occur when someone has a particular phenotype. Now, how can you solve this optimization problem? We searched the theoretical computer science literature, and the work that is most closely related and that we found is the Lightbulb algorithm by Purdue et al. From Colt 1989. They propose an algorithm for the following task. You're observing a set of n light bulbs, which can be on or off. And you're observing them at m points in time. And now what they wanted to find out is how efficiently can they determine the pair of light bulbs, which is most often both on or both off? Or in other words, which is most often in the same state. So they're trying to find the maximally correlated pair, where they define correlation not in the way Pearson did, but rather their correlation score is the probability of being in the same state. Now, of course, you can solve this problem using exhaustive enumeration. You can exhaustively check all pairs of light bulbs in your data set and check how often they are in the same state across your m points in time. But Purdue et al. Came up with an algorithm which can do this much more efficiently. And this so-called Lightbulb algorithm works in the following way. You are given a binary matrix A with m rows and n columns. The n columns represent your light bulbs. The m rows represent your points in time. In our setting, the n columns would represent the SNPs, and the m rows would represent our m patients or plants. And now you repeat the following two steps L times. You sample k rows from this matrix, so you sample a subset of k time points or k patients. And then you sort these, you sort all SNPs based on these on these k patients in ascending order. You can use Reddick sort for that, which means you can do this in a runtime which is linear in the number of rows you're looking at and linear in the number of columns you are looking at. So there's no quadratic effort in here. And after sorting them, you run through this list and you increase a counter for all pairs of columns that match exactly on these k rows. And this whole procedure you repeat L times. At the end of the Lth iteration, you divide the counters that you've collected during these iterations by L and get an estimate of the correlation score of Purdue et al., which is the probability of being in the same state. Now, the amazing fact, which you can look up in their paper, is that with extremely high probability, with a probability of 1 minus n to the minus alpha, this procedure returns the most correlated pair in a subquadratic runtime, in a runtime which is subquadratic in the number of SNPs. I've given it here, this runtime. It's a bit hard to read when you see it for the first time, but the interesting bit is this exponent here. It includes the ratio of the log of P1 and the log of Q2. P1 is the correlation score for the most correlated pair in the data set. Q2 is an upper bound for the correlation score of the second most correlated pair in the data set. And the larger this gap, the larger this difference between the highest correlation score and the second highest correlation score, the lower our runtime for finding the most correlated pair here. Alpha is a precision parameter. You can often set it just to 1. N is the number of SNPs in our setting. I said this is at least 10 to the 5 in our data sets. So the probability with which we return the most correlated pair approaches 1 very rapidly. So it's very much close to 1. There's another question?
Because of the linkage disequilibrium, there will be many pairs, even in the top case, that will have close p-values to each other.
That's a very good point. Maybe I repeat this question for the camera, for the microphone. So the question was that very often there's linkage disequilibrium between SNPs in the genomes, or SNPs are co-inherited, and there may be for instance, several copies of the most correlated pair, and then the difference between the most correlated pair and the second most correlated pair may be very small. Well, in this case, you should not look for the most correlated pair. You should look for a certain set of the top, let's say, H, top 10, top 100 correlated pairs, such that there is a gap in correlation, a significant difference in correlation between these top H SNP pairs and the SNP pairs that follow further down the list. So when you have several SNP pairs that have approximately the same correlation score, then you report more than one, you report 10, 100 SNP pairs until you reach the point where there is a significant difference in correlation between the SNP pairs that are in your solution and that are not in your solution. The same theory holds then for this case. So what this algorithm is based upon is the idea that there is a set of SNP pairs for which the probability of being in this same state is significantly higher than for the rest of the SNP pairs. And this is the set we want to discover using this algorithm. Now, keep that in mind. The runtime for this algorithm is subquadratic in the number of SNPs, and the probability with which we retrieve the most correlated pair is essentially 1 in our setting. Now, this was just a literature review. These were results that we found in this paper by Purdue et al. From 1989. But obviously, this Lightbulb algorithm does not solve the problem that we want to solve when finding the SNP pair with maximal difference in correlation. There are 3 critical discrepancies between our setting and the Lightbulb setting. First of all, we are interested in the difference in correlation. They are just maximizing correlation. Our SNPs, our single nucleotide polymorphisms, are not binary as the Lightbulbs are in a typical setting. And we are interested in Pearson's correlation coefficient rather than the probability of being in the same state. Now in the following, I'll show you in 3 steps how to overcome each of these 3 differences. First of all, we could overcome the problem that we want to compute the difference in correlation, whereas Purdue et al. Maximize correlation. We could prove the theorem that is shown here, that we can define a matrix on which we then run the Lightbulb algorithm, and it returns us the pair with maximum difference in correlation. So how does this work? We assume that there's a matrix of cases A, so of the individuals that have a particular phenotype, let's say a disease. And we have a matrix of controls B of identical size, so with the same number of SNPs. And for the sake of clarity, we also assume that the number of patients in the cases and in the controls is exactly the same. So we have as many cases as controls. And now we do the following matrix operations. First of all, we concatenate the cases and the controls into one large matrix representing all the individuals in our data set. Then we duplicate this data set and concatenate it to the original data set. And at the same time, we flip the entries of matrix B. So far, we still assume that both these matrices have binary entries. I told you before that this is a difference between the Purdue setting and our setting. This is one of the points I listed on the previous slide. Here on this slide, we are just solving the first step. We'll come back to this question of how to deal with non-binary data later. So 1 minus B flips the entries of this matrix. Wherever there was 1 before, there's now 0, and vice versa. And we do the same thing again, just with one difference in protocol, which is we flip A in the second time we do this, and we keep B as it is. And now we could show that if you run the Lightbulb algorithm on this matrix, and then a second time on the second matrix. And when you then report the maximum of the two solutions that you get, then this is exactly the SNP pair which maximizes the difference in correlation between cases and controls. So now we could compute the difference in correlation between two SNP pairs on cases and on controls. What we couldn't do so far was dealing with non-binary data. But this we overcame in the second step. And to do so, we used locality-sensitive hashing by Sharikar from 2002. How does this binarization scheme work? Well, imagine that you are given what I call a SNP vector. A SNP vector would be one column of this matrix. A, for example, it represents one SNP and the states of this SNP in a collection of M patients. Now, as I said before, typically SNPs, in particular in humans, they have 3 states. So this vector is non-binary. We want to make this vector binary. What we do in order to binarize this vector is that we choose a random vector from an m-dimensional Gaussian distribution. So a vector which is of the same dimensionality as our SNP vector. And then we take the inner product between the SNP vector and the random vector. And we check whether the resulting inner product is positive, equal to 0, or negative. If it is non-negative, then we binarize this SNP vector to the number 1. If it is negative, we binarize it to the number 0. When we repeat this whole procedure several times, we get a bit string, a binary vector that represents our originally non-binary SNP vector. And this procedure we repeat for all SNPs, so for all SNP vectors in our data set. Now there's a very interesting fact about this binarization, which helps us greatly to solve the third problem that distinguishes our approach from Paturi's approach. The property is that the hash values of two SNP vectors u and v match with the probability which is the larger, the smaller the angle between these vectors in space. There's a very illustrative way of describing what is going on here. It's a geometric interpretation of what is going on. By choosing this m-dimensional vector r, you are also choosing a hyperplane in your space, because you can interpret this vector r as the norm vector, the vector that is orthogonal to a hyperplane. Now imagine that you have your two SNP vectors in your space, and you choose a random vector r, which defines a hyperplane. Now what can happen? The hyperplane cuts the space into two halves. And now the two outcomes that are possible are the two vectors are in the same half space or they are in different half spaces. When they are in the same half space, they are either both mapped to 1 or both mapped to 0. When they are in different half spaces, then one is hashed to 1 and the other to 0. Now, what is the likelihood that these SNP vectors are located in different half spaces? Well, this obviously depends on the angle between these two vectors. The larger the angle between the two vectors, the more likely it is that a randomly chosen hyperplane will separate the two vectors and put them into different half spaces. This probability of being the same, that is, and this is also very important for us, of course, exactly the definition of correlation that Paturi et al. Used. And now, keeping this in mind, we can now proceed to the third point that we have to solve in order to be able to apply the Lightbulb algorithm to our problem, which is we want to generalize the Lightbulb algorithm to Pearson's correlation coefficient. And now the interesting fact that we can exploit in order to solve this problem is that Pearson's correlation coefficient can be written as the cosine of the angle between two vectors. And now this, in fact, this geometric fact definition helps us solve our problem, because we know now that when the angle is small here, then the probability that these hash codes will match is high. We also know that if the angle is very small, the cosine will be large. The smaller the angle, the larger the cosine. So when we maximize this probability here of being in the same state, and when we maximize the cosine, we are doing the same thing. We are making this angle as small as possible. And this means, in other words, that when we solve the Lightbulb algorithm on the binary data, so when we compute this probability of being in the same state on the binary data, then we are implicitly computing the probability, or not the probability, the Pearson's correlation coefficient on the non-binary data. So when we maximize Paturi's correlation on the binary vectors, we are maximizing Pearson's correlation coefficient on the non-binary vectors. This completes our algorithm. So we have solved all of the three problems I mentioned. And I can summarize now our procedure for finding pairs of SNPs that correlate with a given phenotype in subquadratic runtime in the following manner. Step one is we are given two non-binary matrices A0 and B0. We binarize them into A and B by locality-sensitive hashing. We then compute the maximally correlated pair P1 on this extended matrix that I described before, where B is flipped. This maximally correlated pair we call P1, and we also compute the maximally correlated pair P2 on another extended matrix where we flip the matrix A in the upper right-hand corner. And now in the fourth step, we report the maximum of P1 and of P2, and that's the solution to our epistasis search problem. This is the pair of SNPs that maximizes the difference in correlation between cases and controls. So now, what does this mean in practice? We have a large number of experiments for this. I'm showing a very telling one to you here. We ran this on a data set of 100,000 SNPs from plant genetics. And we adjusted our runtime such that the runtime was n to the 1.38. So we allowed the algorithm to check a total number of SNP pairs which equaled n to the 1.38. And then we looked at the top 10, top 100, top 500, top 1,000 SNP pairs returned by our algorithm after this runtime. And we compared these sets of SNP pairs to a ground truth, which we had computed using our Max Planck computing cluster with 800 cores in Tübingen. So we compared this to a gold reference, and we checked how many of the top 10, of the top 100, top 500, top 1,000 SNP pairs do we recover using our algorithm. And we noticed, first of all, that we were 611 times faster than the exhaustive search, and that among the top 10 SNP pairs, we recovered all of them. Among the top 100, we recovered 86%. Among the top 582, among the top 1,000, 80%. When we increased the allowed runtime to n to the 1.54, we still had a speed up of 97-fold, and we recovered the top 10 and top 100 SNP pairs with 100% accuracy, and even the top 1,000 SNP pairs with 98% accuracy. Empirically, the runtime that was needed to achieve recall rates of this quality here, so close to 1, was n to the 1.5 on our data sets that we work on. Which means that if you have 1 million SNPs, so 10 to the 6 SNPs, you don't have to do 10 to the 12 tests anymore using our method. In the order of 10 to the 9 tests are sufficient now. And this means that using our technique, epistasis detection on the full human genome now would require only a single day on a single computer. This is the theoretical computer science approach to this problem that we worked on over the last year. We also worked, and I mentioned this before, on implementing the exhaustive search on graphical processing units. Graphical processing units were optimized for basic matrix operations for video games, in fact, because updating the state of a pixel in a video game is a highly mathematical operation for which you need a lot of matrix operations, and these graphical processing units are optimized for these matrix operations. Now, when computing p-values, test statistics for the SNP pairs in such a genetics data set, what we need is also basic matrix operations. So graphical processing units are also very useful in our setting, and therefore we implemented our search for SNP pairs that interact with the phenotype on graphical processing units, and we published a series of papers on that and made our approaches more and more general. And in fact, the first one, which computes the criterion that I presented to you before, so this difference in correlation, is already available online. So if you go to this homepage of mine, you'll find the code of EpiBlaster. We've generalized this to non-binary phenotypes recently. This will go online shortly. We submitted a work where we implemented linear regression for SNP-SNP interaction search recently. And all of these will go online over the summer. EpiBlaster, as I said, already is. The Lightbulb algorithm will hopefully follow in the further course of the year. The, for me, highly interesting outlook is the following. Using the Lightbulb algorithm, epistasis search now requires roughly a day on a single PC. Given our GPU infrastructure, the exhaustive search using GPUs also requires roughly a day. Now, the interesting combination which we could do is that we put the Lightbulb algorithm onto GPUs. The Lightbulb algorithm is also only based on simple basic matrix operations. And in this case, we project that we'll be able to perform epistasis search in less than 15 minutes. That would be the hope. That's our goal for the next year, to make this happen. And that would be a big step forward for us, because you needed a giant cluster for several days before to solve this problem. Of course, there's not just a computer science or machine learning side of this problem. There's also a very interesting biological side. And I know this is a machine learning summer school, so I shouldn't spend my whole talk on the biological side. But I want to mention that we not only try to develop these algorithms for finding SNP pairs, we also try to find out if these SNP-SNP interactions that we are searching for actually exist in the data sets that we work on. Because so far, you couldn't really do a SNP-SNP interaction search at a genome-wide scale. So no one could confirm that using this approach you can find interacting SNP pairs that affect a phenotype. So we are entering a new field here now, or a new stage in our research now. Now the goal is, after developing these algorithms, can we get useful biological knowledge using these algorithms? And in order to test this, we run our algorithms on a large collection of genetics data sets from the MPI for psychiatry in Munich. And in one of these data sets, we have very interesting preliminary findings. It's a data set with more than 500 patients and more than 1 million SNPs. And the phenotype is the volume of the hippocampus, so of a brain region. And this hippocampus has been reported to be in connection to Alzheimer's disease before, and conjectured to be in connection with other diseases. And we find several genome-wide significant results here. So we found SNP pairs which are close to genes involved in cell-cell signaling with a p-value of less than 10 to the minus 12. And now our next goal here is to try to find out if we can validate these findings. So there are other data sets for the same phenotype. We try to reproduce our findings in these other data sets. Now there's also a large international consortium, Enigma, that is interested in this problem. And we hope to get more data from them, or we hope to get the collaboration started that help us to verify this finding experimentally, for instance, in an animal model. So the person who is working on this in Munich is Philipp Seemann. This leads me to the end of my talk. 1 hour is over. The short summary of my talk is that using methods from theoretical computer science and extending them to our problem setting, and by using graphical processing units, we now have 2 equivalent approaches for searching the whole human genome for interacting SNP pairs in a single day. And to give you a bit of an outlook to future work in this larger field of machine learning and statistical genetics, a very exciting question, in my opinion, is how to do large-scale phenotyping. I described to you that we now have all these interesting, efficient, and more cheap techniques for measuring the genotype of an individual. I also described to you that we have all these techniques for recording the phenotype. However, extracting the phenotype from these recordings is a very challenging task. So when you have taken pictures of fish, for example, then a human expert has to look at thousands of fish over days, weeks, and months, and has to find out if there is some reoccurring pattern in the appearance of these fish. So this is what has happened in genetics for 100 years now. So in the 1920s, the first guppy researchers sat down and looked at thousands of guppy fish and tried to find special color patterns in male guppies. And they also tried to find out if these guppies somehow differ in shape. And then once they've looked at all of them, they go through them again, and then they annotate each of them and say whether these fish have a particular phenotype or not. So it's an enormous manual effort to derive the phenotypes from these images. Now, our hope would be to use machine learning for this task to automatically extract reoccurring patterns from such a large collection of fish. And Theophanes Karaletzos in my group is working on this problem. Another example for this automatic phenotype extraction is counting leaks, so seed capsules in Arabidopsis. Arabidopsis has all over its branches little seed capsules, and there may be up to 8,000 of them on a single plant. And they are a very good indicator of the fitness of a plant. So the biologists are very much interested in mapping this phenotype to some region in the genome. They want to find out which region in the genome affects the fitness of the plant. But counting all these siliques for hundreds of plants takes years or even more than that. So there's the question of how to do this automatically. And in fact, my student Dominic Grimm, who is here, who is participating in the summer school, he's working on the question whether we can accurately estimate this silique number using machine learning. And for this purpose, we take pictures, different pictures of the plant from different angles, from above, from the front, from the back, from the right, from the left. And then we run some basic computer vision object recognition algorithm on these images to get a rough count for the siliques in each of these images. These counts won't be perfect because the branches, one branch may be in the foreground picture, the other in the background, so the seeds on the background branch is not visible, and the same problem you have when you look at the plant from above. So by looking at a single image, you won't